Okay, my name is Jalen,
My name is Karina.
My name is Andrea.
and we got our project was about Archaea. Some characteristics that make up Archaea are that they are unicellular. They have no nucleus, meaning that they are prokaryotic. Their cell wall is not made up by peptidoglycan. They're made up of other molecules, and they are also known as extremophiles because they are able to survive very harsh environments. Something that we're going to be talking about later on in the presentation. And then to the right side of the screen, it's just the basic anatomy of a prokaryotic cell.
So the taxonomy, the Archaea, classified as bacteria receiving the name Archaebacteria in the Archaebacteria kingdom, and Archaea are further divided into multiple recognized phyla. So we have the evolutionary history. The Archaeans are an ancient form of life. Archaean cells are stomatized dated 3.5 billion years ago, and the remains in lipids have detected in shells dating 2.7 billion years ago. Classifying Archaea remains difficult since the vast majority of these organisms have never been studied in the laboratory or have only been detected by analysis of their nucleic acid environmental samples.
Okay, so Archaea reproduces sexually, which means is independent of any sexual process, versus the binary fission, which is the separation of the body into two new bodies. The organism duplicates its genetic material. Then it's multiple fission. In these, the nucleus first divides into two several equal parts, and their cytoplasm divides into as many cells as their nuclei. And in its fragmentation, the process of breaking off being broken into small or separate parts, and then at last is budding, in which a new organism develops from an outgrowth or bud due to cell division. So Archaea is a domain very diverse, but for me, my research, I found that autotrophs are the most common cells. These produce complex organic compounds like carbohydrate, fats, or proteins. They use these for simple substances that are found in their surroundings, generally using photosynthesis or chemosynthesis, which are inorganic chemical reactions. They use carbon dioxide in the process called carbon fixation, and this involves a highly modified form of Calvin cycle.
Sorry. So the habitats and the environments in which Archaeans live in are extremely hot. Like I mentioned before in the beginning of the presentation, they're known as extremophiles, meaning that they can survive in very harsh environments, such as extremely hot, highly saline, which means they have a lot of salt. They're anaerobic, meaning that they can survive in places where there's very lack of oxygen, and as well as in acidic environments. Examples of these environments would be Yellowstone, which is a park that's very, very, very extremely high. The Lost City would be an example of the anaerobic environment, and hydrothermal vents would also be an example of an extremely hot environment where Archaeans can survive. Archaeans are classified into three categories for the extremophiles. Um, the hypothermophiles are those Archaeans that can survive in very high temperatures. Then we have methogens, which they can survive in places where there's a lot of like lack of oxygen, which would make them produce methane. And halophiles, which means that they can survive in environments that have a lot of salt, which would make them acidic. Particular behaviors of the archaea is their metabolism, especially the methanogenesis. They are able to produce methane by consuming carbon dioxide and hydrogen. Their cell membrane also is made up of ether linkages and monolayers, which is what helps them survive in the extreme environments. And they also have several points of replication, in contrast to other species that only have one point of replication. Species within the groups are Sulfolobus archaea. These are found in volcanic activity, meaning that they can survive temperatures up about to 70 degrees Celsius and with pH levels about 2. Then we have the methanococoides. I don't know how you pronounce it. This is an example of a methogen, archaea. Then we have the haloquadratum waspy. This is an example of a hyperthermophile because it can survive in boiling water. We have the methanosarcina, and they are an example of methogen archaea because they are able to produce methane. And then we also have halobacterials. This is an example of a halophile, because it is able to tolerate high salt environment to live.
Okay, so for the ecosystem, halophilic archaea is a very good example of biodegradation and bioremediation. One of the most important symbiotic relationships within archaea is the ocean and archaea. 20% of microbial cells in the ocean are archaea, and a very good example is green archaeota that scientists suspect may be involved in the marine nitrogen cycle. In the food chains, the archaea is like a lowest level of food chain, and a good example is a producer of metazoan in the Arabian Sea.
Impacts on humans. The Methanobrobacter smithii aids digestion in human intestines. When in the human gut ecosystem, it affects the bacterial digestion of dietary polysaccharides. This influences one's calorie harvest and body fat. Researchers have sequenced M. smithii genome, indicating that it may be a therapeutic target for reducing energy harvest in obese humans.
Some fun facts about archaea is that although they share a lot of similarities with bacteria, they share even more similarities with eukarya genetics. 10% of the ocean life is made up by archaeobacteria, and it is one of the oldest forms of life. And then these are our references that we used. The end.